I've made a really short, really basic talk today, and it's about, a small portion of my research, and main function that I wanted to achieve today is to engage you on some concepts and put us all on the level. But since my talk is quite short, I want to say a couple personal things first. I'm sorry that I missed a couple of the morning talks and the sessions that are going on. I've been in the basement trying to teach genetics to kids through physical activities and music, which has proven to be easily as challenging as my regular scientific research, and I'm probably having less success with it. But we persevere on. Right, and so I'm also really thrilled to be part of a big group of such talented people, and even though I haven't seen everybody's presentations, I've been having a great time interacting and exchanging with everyone. And I'm also very happy to see a lot of familiar faces here. So for me, I've been pursuing sort of in the part-time some projects in science art and science communication. You could argue they're one or the other. It's not really an important point to me. I like to do both. But I've worked in the past with Sasha and Sergey, and we did a theatrical project in St. Petersburg, and that's where I met Dima, and actually where I kind of had the idea that feeds into what we have as the concept for this 9 Evenings Revisited. So I had hoped to do a little bit of a forum or a panel discussion about that with everyone, but I don't want to take up too much time. So I suggest that maybe we get a lot of beers and gather around the tables at some point, and everybody throws out some of their ideas related to the topic, because I'm here to find strong collaborators to work on science art projects. And so that's not, not here to just tell you, oh, this is some stuff I do in the lab, but it's more like I want to connect with you intellectually and figure out what works we can do together in order to make the 9 Evenings Revisited a reality. Right, cool. So with that in mind, we'll jump right into the presentation then. Before I go on, actually, I want to say I'd like to get a round of applause for Candace, who's phenomenally professional organizational skills has made this possible. I can say that being an amateur, my work has been completely amateurish, but now I know what it's like to work with somebody who's a skilled professional, and there's no going back. So thank you, Candace. All right, so I want to talk to you today about a little piece of my work, and these are genetic elements. They're mobile DNA. And in the abstract, I talk about what are called retrotransposons. So they're mobile DNA elements that go through an RNA intermediate. And I'm going to define all of these terms. There'll be a little basic cell biology review to put all of this in context. But what I've put here, just a kind of a summary of different kinds of DNA. So, okay, non-mobile DNA, different varieties of mobile DNA, and the content of those kinds of mobile DNA across different organisms. So, for example, corn, maize, has a lot of mobile DNA, and actually corn was one of the original model systems for the discovery of mobile genetic elements. I'll have one slide on that in a minute. But of course, we're going to focus on man, and man has a lot of mobile DNA, and these retrotransposons, the line elements, what we're going to talk about today, are quite heavily expanded in man, as well as another mobile DNA called the sign elements, which I won't say too much, but I'll try to impress upon you the importance and the enigma of mobile DNA. Now, another point I wanted to make is that, so I'm affiliated with the Rockefeller University, but I'm also affiliated with NYU Medical School. And one thing that's interesting about that affiliation is that I have access to a cutting-edge robotics suite with a lot of really interesting technology. So this is just some playing around. So the group that I work with are synthetic biologists, and some of the stuff that they do is they genetically engineer microorganisms to have particular traits. And a simple trait they might play with is the pigment that that microorganism expresses. So what color is that microorganism? And they have a device which is basically a very small vibrating tip that can dispense single cells or small nanoliter volumes of liquid, so they can print in microorganisms artwork, logos, and so on. So these are genetically modified yeast that have been seeded as single cells onto the surface of an agar plate and that have grown into colonies that display the expected picture because it's invisible when it's printed, right? And so, you know, and this is just playing around, but some of this stuff could be applicable to, you know, media that you are all working with or concepts that you have that we can reappropriate these robots. Yeah. So so that's interesting. So I don't know that because I didn't work on this engineering, but apparently the different colors there are different challenges for obtaining the different pigments. So I'm not sure about the black, but I remember reading about, for example, that purple was a particularly challenging color for them to achieve. Yeah, so this guy here is Gregor Mendel, the father of genetics, who was a monk who did the famous pea crossing experiments. You guys can read about that on Wikipedia if you want to know more, but there's a reason why there's this face here. And of course, you know, for fun, some pop art, right? So I just wanted to like throw that out there, that it's not just that we can intersect on concepts, but we can also intersect or exchange on technology platforms that can facilitate our collaborations. So back to the topic of corn. This is Barbara McClintock, who received the Nobel Prize for her discovery of mobile genetic elements. And actually, she's the mother of our understanding of genetic regulation. So that's usually credited to Jacques Monod, who no doubt was a hugely important figure in establishing how gene regulation occurs. But her work even before then, which was difficult to push through because of the thought leaders of the time, wasn't recognized, but is known now to be the case. So the classes of mobile DNA, right? You have DNA transposons. I had hoped to have a whiteboard here because some of this stuff is easier to draw than it is to explain. But DNA transposons, they go through a cut and paste mechanism, which is basically like there's a gene. Okay, that gene encodes a protein. That protein is capable of excising that gene and moving it to a new location. So they don't expand in copy necessarily, they just change their location. They move from one place in the genome to the next. RNA transposons, which is what we're going to talk about, they undergo a copy and paste mechanism. So the gene produces an RNA, okay, the RNA produces a protein. The protein, instead of binding back to the original gene, it binds to the RNA. It produces a new copy of DNA from that RNA and then inserts a new copy of that DNA somewhere else in the genome. So these elements are capable of rapidly proliferating in large copy numbers in genomes, one of the things that we're quite fascinated about. And what we'll talk about today is this line 1 retrotransposon. Now, in addition to other reasons I'm going to tell you as to why we study these, there is, of course, an importance to human health, which drives a lot of even the basic research investigations I do. So this is a graph that just shows many kinds of cancer. And it shows the percent of patient tissues that show expression of the line element. And the intensity of the color is the degree of expression. So we find that line elements are expressed in over 50% of all cancers. And when they are expressed in cancer, the prognosis for that patient is poor. That means that's a more aggressive cancer. It means that that patient is less likely to recover, even with treatments which are therapeutic. Okay, so let's back up, all right? I like this, matters of scale. Okay, so, you know, we're talking about an organism, a human, okay? And then, you know, I actually had to look these numbers up. This isn't something I would have off the top of my head. So I was like, how many cells are there in the human body? And I looked to see what was known about that. And actually, recently, some new estimations have been done, and they're getting more and more accurate. But apparently, a fair estimation is 37 trillion cells in your body. Okay? And in all of these cells, you have 46 chromosomes. And then in there, you have about 3 billion base pairs of DNA, which, if you unravel and stretch it out, is 1 meter per cell. So you can see how that gets quite long, multiplied by all the cells in your body. All right, now here's an electron micrograph of chromosomes. This is an image that I guess most of you are familiar with because I think that this is in the pop culture. I'm not sure if that's actually true, but it's like I tried to draw this on paper for kids the other day, thinking this butterfly shape would like mean something. Maybe they've seen it before somehow, and so hopefully the same is true for you. But I just wanted to point out that, okay, so in these chromosomes, which are coiled DNA, only about 1% to 2% of the sequence are genes. And the rest, until recently, has been called junk DNA. But, you know, what is junk DNA? I mean, junk DNA just implies that we don't know what it is. We're calling it junk because we don't have a better definition of it. What's that? Superstition, right. So like 2% of it are what we would call a gene. What do we call a gene? A gene is a portion of DNA that encodes the information that produces a protein. So you have a ton of DNA that doesn't produce proteins, and actually about 40 to 50% of that DNA are the result of transposable elements. The line element itself composes 20% of your DNA, and it's the only active retro element. So a lot of those sequences are just like the fossils of a molecular level battle between you and genetic parasites that inhabit you. And sometimes you win out, and then you silence an element, and that element can no longer proliferate. And other times the element finds ways to circumvent the cellular defenses and then proliferate and expand. Now, in our superstitious perspective on these elements, we will call them parasites. They have all the hallmarks of a parasite. We can't identify what they do that's good for us. We clearly see they have great potential to cause and exacerbate human disease, and their only function appears to be, through every lens we've looked at it so far, to expand themselves. But they have co-evolved with us for the entirety of our existence. So if you try to trace back by looking at DNA sequences, accuracy becomes low the further back you go, but these elements are at least 600 million years old, and they probably were with eukaryotes at the divergence of bacteria and eukaryotes at our last common ancestor. So if something's co-evolved with you for that long, it stands a reasonable chance that there are selective pressures or beneficial aspects of that thing that are as yet unrecognized. So just as a side note that I won't talk about here, these elements are normally held to be repressed in your body. They're unleashed in cancer. That's why we see that they're expressed there. But obviously, they're silent most of the time. And that's due in large part to epigenetic effects, which is something you'll hear Sasha talking about. But they're expressed at two key points in the human body. And the first point is in embryogenesis. So when a fertilized egg begins to become an embryo, there's a gene expression cascade that gives way to early developmental biology, and during that time, these elements are also expressed. So at that time, the element is able to insert a new copy into the genome of the developing fetus. And it's estimated that between 1 in 20 to 1 in 40 human births carries a novel insertion of a line 1. So one person in this room, maybe two, have a novel insertion due to this mobile genetic element that neither of their parents had. So there's this idea that it contributes to genetic diversity. And the other cell type is in the stem cells that lead to mature neurons. So it's very hotly debated how frequently this happens, but it is understood that the neurons in the stem cells that form neurons, these elements are expressed and new insertions occur, and that leads to a mosaic in the genome of the different cells in your brain, which, you know, some people want to speculate pretty strongly about higher consciousness. I'm not going to say anything about that today. All right, so like back to basic like molecular biology, cell biology. This is the central dogma of biology, what it's known. You know, what is a gene? A gene is a piece of DNA. This DNA is transcribed into an RNA. DNA is held in the nucleus. We'll get to that in a minute. The RNA is allowed to leave the nucleus, and the function of this RNA is to produce a protein. Proteins are the effector molecules of cells. They carry out all the chemistry of life. Yeah?
Sorry, slightly dumb, but when you say you, do you mean completely unique? Or you...
In the context of insertions in human babies?
Yeah.
Yeah, the location of that insertion is completely unique to that baby. That's not to say that no other baby could ever get the same insertion, but it would have to be a completely independent event that by chance another baby got the same insertion at the same place. Yeah? No, it's cool. It's cool. And so like a side note about that, and I'm talking way out of school here because I'm not a developmental biologist, but the thing is that we can measure when new insertions occur in babies that, of course, are carried to term and can have a sample of their genome taken and sequenced. But since these things are active during embryogenesis, you could imagine, because it's delicate developmental biology, if a new insertion occurs any place in the genome that's sensitive to that, that would then result in a miscarriage or termination of that pregnancy. So I think the true rate of new insertion isn't understood because we don't have those materials to examine to see what's going on with them. But again, that's outside of my expertise. Okay, but the thing is that proteins aren't linear molecules. You know, these are handy-dandy schematic representations of these chemicals that help convey these concepts. But the truth is that proteins fold into very elaborate, exquisite, and beautiful shapes. And their shapes are dictated by their function. There's an interconnection between form and function, and that's one of the things that I'm primarily concerned with. I'm concerned with understanding what are the forms of biological molecules, so then I can match that with their function and understand the connection between form and function, so you can derive some engineering principles. All right, so here we've covered what a gene does. So where does it do it, right? So biochemistry happens in compartments. On the left-hand side, this is just a light microscope image of a standard monstrous laboratory cell line, HeLa cell. This was, I guess, ovarian cancer extracted from a patient quite a long time ago. That is an immortal cell line that we've all worked a lot with, but which probably doesn't really represent authentically like a healthy cell. But it's good for illustration. So you can see there's the nucleus here. That's where the DNA would be held. And then you have this cytoplasm, and all of this is surrounded by an outer cell membrane. So the nucleus has a nuclear membrane, then there's another compartment, and then there's an outer cell membrane. So here, a schematic representation of the kind of biochemical processes that occur is you have the DNA. Now that, of course, undergoes replication when cells split, right, and divide, and each cell needs a copy of the DNA. The DNA also needs to be read to produce RNA transcripts. At least the genes need to be read to produce RNA transcripts. Now we know there are a lot of RNAs that come from non-genes, but I won't talk about that today. Those RNAs undergo processing reactions in order to prepare them for export to the cytoplasm, where they're read by molecular machines called ribosomes, and those molecular machines produce the proteins, and those proteins fold into their elaborate three-dimensional shapes, and those facilitate the chemistry of life. So life is an emergent property of a complex network of interacting molecules. Okay, so that's the crash course in cell and molecular biology. Now, now we can talk about line 1 retrotransposons, right? These guys are crazy. I got into this because since they're not really expressed, right, they're very difficult to study. I became interested in this not only because it's fascinating from a cell biology and an evolutionary perspective. It's that I met people who wanted to study this and couldn't. And what I like to do is develop methods to solve what seem to be insoluble problems. So I decided, okay, this is going to be a model system. I'm going to develop biochemical methods that will allow us to enrich these forms and then study their composition and their functions. So what's pretty interesting about this is actually it's very atypical. Normally you have like human genes, like a single gene encodes one protein. But actually this is a single gene, but it has two what are called open reading frames, creatively named Open Reading Frame 1 and Open Reading Frame 2. You know, super creative. All right, but they're in one contiguous run. So that's very unusual for a human gene. And so what you can kind of do is you can consider this a streamlined parasite. It's not a perfect analogy, but it's like a virus. Like a virus has a compact genome where many proteins are produced by the least amount of DNA possible, and all of those proteins are geared toward ensuring the survival and proliferation of the virus. The difference between this and a virus, or one obvious difference, is that viruses break out of the cell and infect neighboring cells. This parasite has shed the portion of its life cycle that involves leaving the cell. It's happy where it's at, on the couch, watching TV, right? It just goes out there, it snacks from the fridge, and then it goes back in to relax again. So it never bothers to leave. So it produces two proteins, which I'll talk about a little bit, and these two proteins bind to the RNA, and they carry out functions essential for the proliferation of this element. But of course, as any good parasite, it survives by co-opting the proteins that are also present and available in the cell and twisting them to its needs. So the battle, once this thing comes online and these are produced, it's simultaneously stealing your proteins to use them against you, and your cell will begin to produce new proteins to try to counteract that effect. So there's a war going on inside. All right, so what does the life cycle of one of these elements look like? So after they're produced by transcription of the gene in the nucleus, the RNA comes out into the cytoplasm. That RNA will be translated into two proteins, and in a completely bizarre set of events, these same two proteins that are produced from that RNA will bind back to the very molecule that made them. This is very atypical. Normally, a message is translated by the ribosome, that molecular machine that reads the RNA and produces the protein. And then that protein diffuses off to meet with other proteins and form other machines and interact and so on. In this case, the proteins produced by this RNA, because their goal is to turn that RNA back into DNA and bring it back into the nucleus, they are retained at the site of this biochemical reaction, and they bind back to the molecule that created them so that they can chaperone it back into the nucleus and then nick the DNA and then reverse transcribe that RNA into DNA and then insert that new DNA copy. And almost nothing is known about any of these steps. So for me, like, it's something I can play around with, like you play around in the sandbox, and everything you find is completely new. It's very, very fascinating for me. Okay, so just to go through that, right, the DNA makes the RNA. That RNA makes folded proteins. These proteins bind back to the RNA, and then it goes back into your genome, expanding in copy number, all right? And so here's the thing about this, right? So these proteins are produced, they bind back to the RNA, and then, but that's like very abstract. We're like, okay, there are these protein products, they bind back to the RNA, but what does that look like? And plus, we know they bind other proteins in the cell and incorporate them into that particle that contains RNA and protein. RNA and protein particles are referred to as ribonucleoprotein particles because they have ribose, right, RNA, ribose nucleic acids, and protein assembled together. So this is like my artistic representation of what one of these guys might look like. You know, I have the light purple for the ORF1 and the dark purple for the ORF2, roughly put in some kind of a vaguely imagined proportionality that I figure might exist. Okay, so how can we move this out of the abstract and into the real? So I, of course, won't like throw a lot of data points and graphs at you, so I've selected like 2 data slides that are maybe potentially intuitively understandable. So this one is a fluorescence microscopy data slide. All right, so these are cells that we grow in petri dishes, and then we are able to label them with different fluorescent indicators. So then we shine laser light on them, the fluorescent indicator absorbs the light, it emits in another wavelength, and we detect that, and that allows us to define some of the contents of the cell. So on the left-hand side, you see just cell nuclei. We stain the nuclei so that we have a reference for that compartment in the cell, right? I said, you know, there's the nucleus, there's the cytoplasm, there are different structures, and we need to be able to differentiate between those structures. So a way to identify the nucleus is to add a stain that will bind to DNA, and then it will mark the nucleus. Okay, now we have an antibody against this protein ORF1. If you're not familiar with what antibodies are, I mean, everybody's heard the word an antibody. You know, you become infected, your immune system produces antibodies. Well, one of the things that antibodies do, what they're great at, is binding to proteins. So if you get foreign proteins in your body, part of your immune system reaction will be to produce antibodies which can bind to those proteins and sequester them for cellular processing reactions, okay? I won't go into that. But we can reappropriate those tools in order to identify proteins we're interested in. So we have an antibody against ORF1. So if this is the nucleus, we can plainly see that most of the ORF1 is in the cytoplasm, right? The nucleus is dark, okay? Now if we stain for ORF2, we can get 2 different kinds of staining patterns. We can get a staining pattern where it's in the cytoplasm, and we can get a staining pattern where it's in the nucleus. So there are distinctive portions of the life cycle of the particle that are in the cytoplasm and are in the nucleus, but it appears that it's probably only ORF2 that goes into the nucleus, but both ORF1 and ORF2 are in the cytoplasm. So if we look at the merged photo, that might become more clear. In the merged photo, you can see cell nuclei, right? They're in blue. Now, in the red is ORF1. So you can see that all of the cells express ORF1. And in the green is ORF2. So cells which are yellow, right, it's a green-red overlay, they're expressing both proteins, and we can see them both in the cytoplasm. And in some populations, we can kind of see a strong green in the nucleus. Now, I can't rule out that there's not like a small amount of ORF1 in the nucleus, but it's all about comparing the relative levels. So what's the intensity of the hue in one compartment or another? It gives you an indication of the residency of that molecule in that cell compartment.
John,
Yeah.
maybe you mentioned.
Yeah.
What's the difference between the top layer and
Oh, they're just two different sets of slides, right? Okay, so I wasn't going to get into this, but for the example to show that when we see dividing cells, we see neighboring nuclei stained for ORF2. So one model we're working on is that ORF2 gets into the nucleus during mitosis, right? But that's something we're still exploring. So we have one field where we have, you know, just some cells that aren't paired, obviously, that are expressing ORF2, and then we have these cells which are opposite that have the nuclear staining. All right, so another method that I can use to characterize particles also takes advantage of antibodies. So this is me killing a cell, right? I mean, I kill them in the billions all the time, but apparently that's not murder, right? And what I do is I release their contents, right? Because I want to examine their contents, I have to let them out. Now, the challenge of that is to release the contents in such a way that I don't also change the contents while I do that. And there's a lot of technology and specifics behind that I won't go into, but suffice to say, all the kinds of lines of investigation have different potential artifacts and different potential information you can gain. So on the one hand, I might want to look in cells using fluorescent markers. On another case, I might want to break the cells open, release their contents, and study those contents. So I can use antibodies in the same way to enrich on an insoluble medium the target protein. And that's a protein I know. I have an antibody against a protein I know, and I can enrich that protein I know on that insoluble medium, removing it from a cell extract, from solution. But as a consequence of immobilizing that protein, other proteins I don't know yet, who associate with the protein I do know, guys who are guilty by association, will come out of that mixture, out of solution, and be immobilized. And then I can later release those and separate those molecules into individual species. So an example of what that looks like is this. This is you know, a schematic representation of that experiment, and this is real experimental data. So this is when I use an antibody against ORF1, right? And that's ORF1 at the molecular weight for ORF1. And this is when I do the same experiment for ORF2. This protein here is ORF1. And then each of these bands represent a different protein that also interacts with ORF1 or ORF2 or both. And I can identify those molecular species using different analytical strategies. So what I'm doing is I'm building up a list of the other proteins that participate in the life cycle pathways of line 1, right? And then that helps me, let's say, test my hypothesis about what the configuration of proteins are in this particle. Okay, so enough data. Just I want to recap to you the concept overall. So we have in our cells 100,000 copies, roughly 20% of our genome is a genetic parasite that's been proliferating within our genome over our evolutionary history. In every individual in the room right now, about 100 of these loci in your genome are potentially active, can produce a transcript, can produce a retrotransposon, and can create an insertion. That insertion could not do anything, or it could damage an important gene and lead to disease. And these insertions, of course, happen exacerbating cancer, they happen in embryogenesis, and they happen in neuronal stem cells. So this guy has a life cycle where he's transcribed, producing an RNA molecule. That RNA molecule is exported to the cytoplasm, where the protein products of that RNA bind back to the RNA. I didn't go into it, but based on the data we have, it looks like there are many more copies of the ORF1 than the ORF2, so that probably participates in other additional complexes that I'm examining in other lines of investigation. These guys are translocated back into the nucleus, where a process, what's called template primed reverse transcription occurs. It's not worth getting into the nitty-gritty of that, but the bottom line is that this ORF2 protein is able to nick the DNA, and then it's able to produce a complementary DNA copy of the RNA it's holding onto, and then insert a new copy into the genome. So that's the life cycle of a streamlined DNA parasite. So with that, I'll thank you for your attention, and of course I thank the places I work, the Rockefeller and NYU, and I want to thank the Kunstkraftwerk for hosting us. And in case you guys are interested, you can take a look at, this is the National Research Center I'm affiliated with, the National Center for Dynamic Interactome Research, and we're relaunching our website soon, but you can still take a look at the current one. And it's a whole network of researchers who develop different lines of technological and methodological investigations, solving tough problems in cell biology, biochemistry, and systems biology. And if somebody else's work interests you, I'd be happy to introduce you to them, because this isn't, as much as I want to make productive connections and collaborations for science and art, I also want to connect artists with any scientists they might be interested in having a discussion with. So please don't be a stranger.